Western blot is a technique used to detect and quantify specific proteins within a complex biological sample. This assay involves several steps including: first, sample preparation and protein extraction. Second, protein separation via gel electrophoresis. Third, protein transfer from the gel to a membrane. Fourth, incubation with primary and secondary antibodies. Finally, detection of the target protein and recording protein bands. Then the target protein expression is normalized relative to loading control or housekeeping proteins such as beta-actin. Regarding sample preparation in Western blot analysis. First, the tissue sample is dissected out, and lysis buffer such as RIPA buffer is added. The lysis buffer contains protease inhibitors to prevent degradation of target protein by endogenous proteases released during cell lysis. Also contains phosphatase inhibitors, to ensure integrity of phosphorylated proteins during cell lysis. Finally, homogenize the tissue in the lysis buffer, then centrifuge at 4 degrees Celsius. Then collect the supernatant and discard the pellet or debris. Regarding protein quantification in Western blot analysis. Use a colorimetric assay such as the Bradford or BCA assay for protein quantification via the following steps. First, using standard protein with known concentration, prepare serial dilutions and obtain the corresponding absorbance using spectrophotometer to generate standard curve. Second, measure the absorbance of each sample and use the standard curve equation to obtain the corresponding protein concentration. Use an aliquot containing known protein concentration, such as 20 microgram protein, from each sample. Then this aliquot is loaded with equal volume of loading Lemley buffer that contains sodium do-decyl sulfate. SDS acts to denature proteins and coat them with uniform negative charge. Afterwards, heat at 95 degrees Celsius for 5 minutes to ensure protein denaturation. The importance of protein denaturation and coating with negative charge is to prevent variation in molecular shape and charge from affecting protein migration pattern in the gel. So denaturation and coating with negative charge ensure that protein migration and separation will be only according to protein size, with smaller proteins migrate more quickly through the gel. Gel electrophoresis and protein separation. Polyacrylamide gel is used, and placed into the electrophoresis tank. Fill the inner and outer chambers with electrophoresis buffer. Then load the protein ladder containing a mixture of proteins with known molecular weights in one lane. Afterwards, load each sample into a separate lane. Run the gel in the electrophoresis buffer and monitor the tracking dye, such as bromophenol blue. Stop when the dye reaches the bottom of the gel. Then, turn off power and remove the gel carefully from the tank. Membrane transfer or blotting. After protein separation on the gel, the protein should be transferred to a membrane made of PVDF or nitrocellulose. To assemble a transfer sandwich, place the gel next to the membrane, with filter papers and sponges on either side to ensure a secure fit inside the cassette. Then ensure that everything is fully wet with the transfer buffer inside the tank. Afterwards, a current is passed through the sandwich to drive the proteins from the gel to the membrane. Blocking the membrane. Before the target protein can be detected, the nonspecific binding sites on the membrane must be blocked via incubating the membrane in blocking buffer containing dry milk or bovine serum albumin. Primary and secondary antibody incubation and detection. The blocked membrane is incubated with the primary antibody overnight at 4 degrees Celsius to ensure binding of the primary antibody to the target protein, then wash the membrane with TBST buffer. Afterwards, incubate with horseradish peroxidase conjugated secondary antibody against the primary antibody, then wash the membrane with TBST buffer. Then the detection. Horseradish peroxidase enzyme reacts with a substrate, giving color or chemiluminescent signals recorded as protein bands using imaging system. Quantification of protein expression. The captured image of the membrane is open in image analysis software, such as ImageJ, to measure the band intensity. Taken in consideration that the housekeeping protein, or loading control, is beta-actin. Calculate normalized expression of the target protein by dividing the target protein band intensity by the beta-actin band intensity. Note that both numerator and denominator are arbitrary intensity values, so the units cancel out, and the final result is unitless. Therefore, data are presented as normalized relative expression with arbitrary units. To sum up, Western blot is a technique used to detect and quantify specific proteins within a complex biological sample. This assay involves several steps including: first, sample preparation and protein extraction. Second, protein separation via gel electrophoresis. Third, protein transfer from the gel to a membrane. Fourth, incubation with primary and secondary antibodies. Finally, detection of the target protein and recording protein bands. Then the target protein expression is normalized relative to loading control or housekeeping proteins such as beta-actin.